Good afternoon, everyone. So in the afternoon session, we have Dr. Donald with us. So those who don't know him, like Dr. Donald James, is recently joined as Scientist B in the Biotechnology Department of the Institute. So today he will be speaking to us on the art and science of genome editing. So Dr. Donald did his MSc in molecular biology and biotechnology from IARI, New Delhi, and also completed his PhD from International Center for Genetic Engineering and Biotechnology, New Delhi. After that, he was awarded with SERB National Postdoctoral Fellowship in Genome Editing, and he was attached to CCMB, Center for Cell and Molecular Biology, Hyderabad, for his postdoctoral program. And he has more than 15 research publications in highly reputed journals to his credit. And after joining the institute also recently, he was awarded with the highly prestigious Marie Curie postdoctoral fellowship. So I think like he's an apt person to deliver a lecture on genome editing. So with great pleasure, I would like to invite Dr. Donald James for the lecture.
Thank you, Dr. Suma, ma'am, for the introduction. Good evening, friends. I'll be talking on the art and science of genome editing. So before we go for the main talk, I have divided my talk into certain sections. So what is genome editing? A brief history. How does it work? Why should we edit genes and genomes? Some success stories and applications, and the ethical and ecological issues surrounding genome editing. So before we go ahead and talk about genome editing, I'll just refresh. So for all those people who are not from a pure biological background, there may be some of you. So we'll just discuss about what genomes are, what genes are, what is DNA, what is chromosome. So basically, if you see, every cell of our body, of any eukaryotic organism, will have a nucleus. And in that nucleus, you will find the genome of that organism. And genome is defined as the complete set of all genetic material of an organism. And it is packed very tightly into this small space of the nucleus. And in fact, it's so tightly packed that if I stretch the nucleus, the DNA of an adult human man, I'd get the size of my height. And if I arrange those from the 10 trillion cells in my body, if I put them on back to back, it can reach the moon almost 6,000 times. So that's the amount of packing. You can imagine that the DNA is so well packed within a small size of this nucleus. And so once the whole set of genome is packed into chromosomes, and within that, you will find the 2 nanometer thick DNA double helix. And this is the genetic material of almost all the organisms we know of, except for maybe you will already know coronaviruses are RNA viruses, and they have an RNA genome. So you can ask me whether only the nuclear component is called a genome. No, but nuclear component is not the only one which is called genome. Even the genetic material present in chloroplasts as well as mitochondria are also included in the definition of genome. So then you can ask the question, what is genome editing? So basically, we have a technique in which we are able to precisely modify DNA in the genome in vivo. So that is the key word, precisely and in vivo. So till now, you can ask me, what is the difference between genome editing and genetic engineering? So genetic engineering has been a mainstay in science from 1970s onwards. But if you compare genome editing, it is a recent technology. It has been just after 2012, you can say it has kicked off. And what is difference is that earlier on for genetic engineering, we'll have to take the DNA out into test tubes or into small vials in vitro, do the manipulation, and then put it back. So that is the difference between genome editing and genetic engineering. In genome editing, you're able to do that in vivo within the cell of a living organism, right? And so basically, we're making specific DNA cuts using things called engineered nucleases. And through this, we can add, remove, or alter the gene or multiple genes of a genome in any organism. So that is the beauty and the revolutionary technology called genome editing. And by this, by changing this genome, we can change the characteristics of a cell and also through that, the traits of an organism. So we have had different techniques available to us throughout the years for genome editing. But when I said from 2012, it was due to the discovery of something called CRISPR-Cas. And I think most of you must have heard either through the newspapers or maybe through science articles of the revolutionary technique called CRISPR-Cas. In the first generation of genome editing, we have had nucleases such as zinc finger nucleases, transcription activator like effector nucleases, also called talons, and also meganucleases. But these are all included in the first generation of genome editing. In the second generation of genome editing, it's a revolutionary and more precise technique compared to these. And that's what I'll be talking about mostly in this talk. So what is CRISPR? So CRISPR is an acronym for the clustered regularly interspaced short palindromic repeats. And basically it may look very like a big thing to learn or something, but it's not that a great deal to understand this. So basically Francisco Mojico from the University of Spain was the first person to give this name. But initially the sequence itself was discovered by a Japanese scientist out of University of Osaka in Japan. His name is Ishino. So I'll get back to Ishino in a while. But Francisco Mojico was the first person to name CRISPR as such. And so basically CRISPR is a family of repetitive DNA sequences found in bacteria. And Cas9 or Cas is the CRISPR associated protein. So these are two components involved in genome editing. One is the sequence itself and one is the protein. And CRISPR consists of around 20 to 37. So 20 to 37 is a wide range. And because depending upon which bacteria it is taken from, it differs. And it consists of 21 to 37 base pair direct repeats interspaced by similarly sized non-repetitive variable sequences called spacers. So basically in this figure, you can see there are repeats. Basically what a repeat means is the same sequence occurring again and again. And in between, there are regions called spacers, which have no similarity in between each other. So this is the first report in 1987 of CRISPR sequences. It was not named CRISPR at that time, but the Dr. Ishino from the University of Osaka in Japan, he was studying a gene called IAP. So basically, he didn't have anything to do with CRISPR. He didn't know the profound implications of such a technology at that time. But he was very keen on making succinct observations. So this is an example of how, even though you're working in another field or something totally unrelated, the observations which you make in science can be utilized by others for making great discoveries. So that's why the importance of keeping records in science, as well as the importance of noting down and reporting whatever we observe in the field is very important. So this is one such example. So in his own words, he has said that he didn't know the biological significance of this sequences. It is such an oxymoron saying that he didn't understand what this could become in later years. So this is the first report, and he saw that there were repeated sequences. He didn't understand them, but he reported them. Later on in 2000, this person which I talked about, Dr. Francisco Mojico, he's another something which we can also learn from him. He was the first person to actually give the idea that CRISPR is involved in the immunity in bacteria. Till that time in 2000s, we were of the opinion that most of us, only humans or maybe animals, have an immune system, not even plants, right? So this is a totally absurd and it was what do you say? It's a totally new thing which he was putting forward. And in fact, he was sending his papers to Nature to PNAS, to Science, and all of them rejected him. And eventually it was published in some obscure journal. But later on in 2005 and in 2007, another group of scientists who were also not working on CRISPR, they were working on something called Streptococcus thermophilus. So Streptococcus thermophilus is used in the yogurt and cheese industry. And they were working in a company called Danisco, which was making sure that the Streptococcus which they were using do not fall prey to bacteria, to viruses. So I'll ask you a question. Do you think bacteria ke pani pidikkyo? Arkeloreo? Not in the strict sense of the word fever, but do you think bacteria are infected by viruses? Yes. So can you tell me of any name of the particular virus which infects bacteria? Yes. So bacteriophage, most of the biological students will be okay with this. But so basically what this is a bacteriophage. So like coronavirus infects our cells and uses our machinery to make its progeny. The bacteriophage uses the bacterial cell, in this case, especially Streptococcus, to use its cell hosting machinery. It will, through it will replicate its genetic material and it will create progenies. So in the first case of infection, when a bacteriophage infects the bacteria, Streptococcus in this particular thing, what it does is the bacteria will take a small part of the viral genome and keep it. So that's what you see here. As soon as the bacteriophage infects, it inserts its genetic material. In the normal process, if it was not resistant. So what these people saw that when they were checking for resistance, they saw that one lot of their Streptococcus thermophilus was resistant to the bacteriophage. That means it was growing even though it was infected. So they wanted to know what is the reason behind that. And then they saw that, so that is how they had to take painstakingly, they dissected the reasoning behind that. And they showed that for the first time that as soon as the genetic material of the bacteriophage is inserted, a small portion of that, what we call as spacer from this region, from the previous slide, that spacer region is incorporated into the genome of the bacteria, a small portion. So this small portion, it has very profound significance, which I'll talk to you about later. But within bacteria, there is something called a CRISPR locus, where as soon as the bacteria recognizes a virus, it just cuts and cleaves some small portion of that as a memory. So basically what we are seeing is what do you say, an old-fashioned way of immunity in bacteria. So it also has a Cas gene cluster, an operon, and it produces what is known as Cas9. So these two things you should keep in mind. One is the CRISPR sequence and the Cas9. So when the second time it is infected by the same virus, what it does is it will recognize that this is the same virus. And because of that, it will release it will make RNA molecules out of the spacer which it has kept in the previous infection. And this Cas9 is a nuclease. So it will go and bind to this, it will recognize this foreign DNA which has come from the second infection and cleave it. And therefore the virus is now dead. So basically, this is how they have shown that this is the CRISPR-Cas9 system is involved in adaptive immunity in bacteria, right? So what does that mean to us? It doesn't mean much. Bacteria what do you say, immunity system under. But the ingenuity of human mind is that they, two of these people whom you see here, Jennifer Doudna and Emmanuel Charpentier, independently were working on studying RNA, right? And they came to the conclusion that, okay, if this is cutting viral genomes, what if we can engineer this in such a way that it can also target any genome? So this was the ingenuity, and they published this in Science in the year 2012, in June 2012. And their ingenuity was that they combined two different aspects into one and made it simpler. So that is the key point. I'll not go into the details because most of you are not from this field. So, and they got the 2020 Nobel Prize in Chemistry for this Normally, it takes a lot of time for getting a Nobel Prize. Most of you would know. It's not given out very lightly. But the implication and the revolutionary nature of such a tool was crucial in them receiving the Nobel Prize so early. So how does this CRISPR work? So basically what you're seeing is, as I told you, there are two main components in the CRISPR system. One is the Cas9, the nucleus, which cuts the DNA, and the other is the guide RNA. So the guide RNA is the one which targets it to the specific region in the genome where it has to do its work. And the Cas9 is the nucleus which will cut it. So this is what is happening over here. And once it is cut, then people can do different things with that. So the CRISPR-Cas9 based genome editing. So now we know that there is a CRISPR system. It was a bacterial immune system, and it can be repurposed in such a way that we can target any genome in any organism. So how does that work? So first of all, we need to design what is called a single guide RNA, which is actually the targeting molecule. And the other thing is the Cas9 endonuclease, which is the nucleus which will cut the DNA. And once it cuts, there are four options for the organism to, it can go through any of these four ways. It can be perfectly repaired by the repair mechanisms within the organism. So there are DNA repair mechanisms within humans, within different organisms, in which any changes to such mutations can be repaired. But in some of these cases, there is a way called the non-homologous end joining. I'll not go into the details. So if it goes through that, there is a mutation coming up here, which will may cause the gene to be functionally invalidated. So in that case, what we call is, it's called SDN1, and that is a knockout, a gene knockout happens. In another case, you can give a template which will replace a small region in that gene, and you can actually, we can go for an alteration in that gene. So this is called through homologous recombination, this happens, and this is called SDN2. And in totally different, we can actually go and add a foreign DNA through this process, and that is SDN3, and that also happens through homologous recombination. So there are various ways in which this is being applied at this level. We can go for, I won't go into the details, we can go for gene knockout, means you can functionally stop that gene from working. You can also maybe put in a functional copy of a gene which is not working, right? Or we can replace a specific region which is mutated. We can also go for base editing. So in that video, it would have been very clear for you. But for those who don't know, DNA composes of 4 different bases, ATGC. And it is a run of all these bases. So if you want to change only one single nucleotide at the base level, you can change that also by combining this Cas9 with different types of enzymes, right? So Cas9 is now not cutting, it is just in the process of bringing this specific enzyme, another enzyme, to that region, okay? So that is base editing. Then there is also, we can go for RNA editing at the RNA level. It's not at the DNA level. We can also go for increasing or decreasing the expression of genes using CRISPR-I or CRISPR-A approach. We can go for epigenetic modifications. I'll not go into details of that also. We can also use CRISPR to visualize where in the genome a specific sequence is. So you just have to combine it with different molecules, fluorescent molecules, to visualize the genome, right? So what is the reason why we do all this? So why are we doing all this? Why should we go for genome editing? Why should we go for editing a gene? So basically, this is the central dogma, which many of you biological students would know. In every cell of ours, right now, as I speak, almost in 1 second, 20 million cells of RBC is being produced. And although nucleus is not present in RBC, but every time it is being to be produced, the cell has to divide. And every time a cell in the human body or any organism divides, it has to undergo replication. The genetic material also has to be replicated. And since when this replication happens, and this is called the central dogma because this DNA is controlling the formation of an RNA, And from that RNA, through transcription, and from that RNA, a protein is made. And so you can see that all the whole body, if you see, our skin, our bones, they're made of collagen. Our hair is made of keratin. If you see the plant material, rubisco is the most abundant protein in the whole biosphere. So there is protein all around us. And all of this is being made by this process, right? And the main issue is that whenever the replication fidelity is having a problem, it leads to something called a mutation, right? And due to this mutation, it will form what is called a mutated protein, an aberrant protein. So when this aberrant protein is formed, it can lead to cancer and diseases in humans. And if this mutation is occurring in the germline, that is, it is being given to you by your parents even before you're born, what happens is that we get different kinds of diseases called monogenic diseases, genetic diseases, which are carried on. And there are many of them, but I'll tell you, these are all monogenic. That means just one single gene is causing such a drastic change in our phenotype. One very common one is the sickle cell anemia. So sickle cell anemia, if you know about sickle cell anemia, the RBC is taking a shape of a sickle, Arival's shape. When this happens, oxygen consumption by that person is drastically reduced. He cannot run, he cannot do anything, and life becomes miserable, right? So, they can only live if it is in a heterozygous condition in which at least one allele is normal, right? So this is one single, basically just because of one amino acid change in the hemoglobin protein, just one amino acid. And even in other cases, such as Duchenne muscular dystrophy, there is only one single, there is a mutation in one single gene. And similarly, in osteogenesis imperfecta, what we call as brittle bone disease, it is just due to one change in the collagen gene. And so this has got profound implications. And there are similar different kinds of single monogenic genetic diseases even in animals. And when you come to plants also, although those which are having genetic problems will not survive and will not care about them because they're plants. But when you want to grow a crop as such, and it is being infected by diseases, then it is a problem. And how much of a problem it is, you can understand by two different famines which happened all totally different places from across the world. One is the Irish famine of 1840s. It wiped out nearly 25% of the Irish population at that time. Okay, and that was caused due to late blight caused by a fungus called Phytophthora. And also the Bengal famine, in which 2 to 3 million of our compatriots perished. It was not only due to the brown leaf spot of rice, which was the causal organism of that, but it was also due to some socioeconomic conditions at that time. But still, the brown leaf spot was one of the major reasons. So right at that time, we only had, and even now, most of our focus is on breeding techniques. We just cross, so we see some resistance somewhere, we cross it to another plant, and you put that resistance into that plant, and that is how we are doing right now. But there are cases in which you will not find resistance in the germplasm. So what do you do for all that kind of diseases? So you can see that just because of one war in Ukraine, prices have soared for wheat and all. So similarly, if there are conditions inside, like we are having COVID pandemic, plants also have pandemics. And so this is something of that sort. And there are susceptibility genes, which we now know of, which we can mutate and we can make it resistant. So that is the beauty of using CRISPR or genome editing to prevent such kind of issues. So other than that, CRISPR can also be used for functional studies. So if you want to understand a gene, if you want to understand what it does, we may not be able to work on humans as such because it is unethical. It's a simple matter of fact. But because of something called collinearity, the genes order in a species such as a fly. നമ്മുടെ പഴത്തിലൊക്കെ വരുന്നിരിക്കുന്ന ഈച്ചയില്ലേ, ഡ്രോസോഫില എന്ന് പറയും. അതിന്റെ അകത്തുള്ള ജീൻസ് ചിലതൊക്കെ it is very similar in the structure, in the order to what is present in a human when it is being developed in the embryo. The positional effect. So this is what we call the Hox gene family of homeobox proteins. I'll not go into details. Basically, ഒരു സ്ഥലത്ത് ഏത് അപ്പൻഡേജ്, ലിംബാണോ കാലാണോ കൈയാണോ എന്താണ് വരുന്നതെന്ന് തീരുമാനിക്കുന്ന പ്രോട്ടീൻസിൽ ഒരു ഫ്ലൈയിലും ഒരു ഹ്യൂമനിലും ഭയങ്കര സാമ്യതയുണ്ട്. അത് കാരണം തന്നെ നമ്മൾ ഫ്ലൈയിൽ പഠിച്ചു കഴിഞ്ഞാൽ ഹ്യൂമൻസിലും അത് കുറേയൊക്കെ ആപ്ലിക്കബിൾ ആണ്. So that's how we can distinguish and differentiate and study the function of different genes in different organisms. So basically why we use a fly is because this fly specifically Drosophila is a model organism. And most of the biology students would know why we use model organisms. So model organisms are very fast breeding. So within 10 days, the life cycle of this fly is over. So we can study generations upon generations in a short period of time, right? And also this is very small and it is easily growable in a lab. Similarly, in a case for plants is Arabidopsis. So Arabidopsis and Drosophila are, even C. elegans is another model organism. So the reason why we study is because we can study a large number of generations within a short period of time, and we can easily maintain these in the lab. So also CRISPR is now used for diagnostics. So basically because it is binding to a specific nucleic acid, and most of the viruses and the diseases contain genetic material as such. If you want to know what is infecting a person, we can use CRISPR for that diagnosis. In fact, CSIR IGIB, Institute for Genomics and Integrative Biology in New Delhi, have released in association with Tata Group a kit called Feluda kit. Basically, it's an acronym for FN-Cas9 editor-linked uniform detection assay. And this is already available. This was India's first CRISPR-based kit for diagnosis in COVID time they had released. So other than that, the main thing I think for CRISPR is for use in gene therapy. So as I told you, cancers can come because of such mutations in germline or during this, when we are attacked by radiation, mutagens, cigarette smoke, whatever. So those kind of mutations can come, and we want to find out an efficient way in which we can, in that generation itself, in our bodies, we can do the edit, right? So this is one way in which we can use CRISPR for gene therapy. And there are successful stories out there for gene therapy. In fact, so CART cell therapy, basically chimeric antigen receptor based therapy, is already well known. But now CRISPR is acting as a, an increase in the efficiency of CART cells. And basically, CAR cell-based therapy is now working. We know it is working because a lymphoma patient, he was in the trials, he's still surviving without remission in the 11th year. And for acute lymphoblastic leukemia and such diseases, there is no cure other than gene therapy. And CRISPR will enhance the efficiency of such therapies. A recent report also shows that CRISPR was helpful in providing eyesight to a person who was blind. And she was infected with, she was suffering from what we call a congenital disease, Leber congenital amaurosis. And they were able to increase her sight. And this is, even now it is in the infancy stage, but more and more improvements in the CRISPR technology will further lead to, although a few of the patients it doesn't help, but because of rapid improvements in the technology, it may be in the future that most of these cases can be treated. Also as a cure for AIDS. So HIV can be eliminated from the genome using this. So this is something which I have been working on. I have been working on a system called Xanthomonas, which causes bacterial leaf blight in rice. So in this case, what bacteria does is it will infect the plant. It will cause the plant's sweet gene which is a gene which is responsible for transporting sucrose out into the xylem. Because the xanthomonas lives in the xylem, it will cause that sweet genes to increase expression using something called tails, and that will cause, because of the high amount of sucrose and the fructose in the xylem, the bacteria can survive. And this is causing some very 70 to 80% of our what do you say, the yield of rice is decreased because of this this bacterial disease. And so if you are able to knock out that specific region in the genome where these tails are binding, you can prevent this from happening. And that's exactly what they did. They published it in Nature Biotech, and it is now a resistance mechanism, right? Similarly, something trivial as such as non-browning in mushrooms. So if you cut an apple or if you cut a mushroom and you keep it outside for some time, you will see it is browning, right? And that is because of action of a gene called polyphenol oxidase. So if because of this, it is causing polyphenol to oxidize, and then that oxidation products are what you see as browning. Now, if you're able to knock out this PPO gene, and that is what they did using CRISPR, we'll get white mushrooms which will have a higher shelf life instead of it being thrown out very fast. Similarly, this Arctic non-browning apple, although it was made from the RNAi technology, which is a different technology rather than CRISPR, this can also be, CRISPR can also be applied to similar for making such apples. Now, one controversial and very controversial technique, which has been a derivative of CRISPR, is the gene-derived technique. So basically, what is it is that Oxitec is a company based out of Oxford, and what they have done is they have made a transgenic mosquito, which has got a self-limiting gene. So if a wild male, if a Oxitec transformed male mosquito mates with a female wild type, what happens is the progeny dies because of the CRISPR drive or gene drive. And this is basically, it's causing it to extinction, right? So there are ethical issues behind this, but people are going, so the state of Florida in US has already gone forward with this, but it raises a large number of questions. So are we allowed to play God? Are we allowed to cause the extinction of a species? So will this small, what do you say, because of the effect of the death of some mosquitoes, will it cause a ripple effect in the ecosystem? Was the mosquito playing some kind of role that we don't know of in the ecosystem? And also, we know that insects can have pathogenic. So basically, is it a driving force for such kind of insects to turn into pathogenic insects? So these are all the questions which are raised by such kind of these things. There are also an ethical question of germline editing. So this was something which was very, very controversial. In 2018, a Chinese scientist, He Jiankui, he edited and created the world's first CRISPR babies. So basically, his intention was to edit this CCR5 delta gene, and that will lead to HIV resistance. But what happened was that he did it in a way in which all of these genes, which the gene which he edited, he did not report to his seniors. He did not report. He did not give correct details. He in fact also faked a test. So the unethical way in which he proceeded with these experiments raised a lot of problems, and he was jailed. And only last month he has been released from jail. And because germline editing and creation of germline edits are not allowed in any countries, even including China. And also, ironically, the specific gene which he targeted, although it was for a good intention, it later was found that it also leads to 21% increase in mortality. So basically what he did was doom these kids, although he wanted to save them, because we do not have all the information before us. If we jump into something which we don't know about, it may lead to unintended consequences. And this is a prime example. And this was the first and the most significant example of such thing happening. And we do not have an undo button for what we have done. That is the most crucial aspect of that. So with great power of such kind of technologies comes great responsibilities. And although we can go ahead with targeted medicines, biofuels, disease-resistant crops, new industrial products, but the safety, the ethical concerns, and the regulation are very prominent questions which are before us, and especially germlining, which I talked about right now. In Nazi Germany, there used to be something called eugenics. Basically, people are selecting based on how they look. So basically, you're creating supermen and superwomen. So that's a very unethical, what you say, a way to see the world. And also, people can use this technology for bioweapons. And also, it will, if you are using gene drive and such for other organisms, it can lead to biodiversity issues by reducing the biodiversity. And the biggest problem of CRISPR, which I had not mentioned till now, is the fact that even though it is very precise compared to whatever technologies we had before, CRISPR also has some problem, as it can go and edit something somewhere else, which is called off targets. So if that happens, it is causing much more problems. So recently, India has deregulated the CRISPR, and it has considered SDN1 and SDN2, which I talked to you about earlier, as we can release them as varieties now. So we do not have to see them as transgenic crops anymore. So this was just last month this has happened, right? So this also raises a lot of questions. Are we ready for this technology? Is it worthwhile that we do this? And what are the unintended consequences of such a technology? And who is regulating, and how much of regulation is there? So all these are unanswered, and many of them are being answered through our governments. But still, we need to also, as the public, we also need to be involved in this, because it also affects us. And there is another aspect of the cost and accessibility. So Zolgensma is a gene therapy for what we call spinal muscular atrophy. So you must have seen a lot of celebrities in social media pleading for money to save people, so save children who are affected with SMA, right? So Zolgensma is a gene therapy which is the world's most expensive drug. For one single application, it costs 16 crores, right? So one single application can save that child's life to an extent, maybe 70% accuracy, maybe can save that child's life. So this Zolgensma, although it's not CRISPR based, because of the advent of CRISPR, people can go for gene therapy based on this CRISPR techniques. And then the thing comes is, what the question next comes is, what about the patent? So this is Feng Zhang from MIT. He was the first person to actually show that this technique can be done in human and mouse cells, to actually edit it. Although it was proposed earlier on by Doudna and Charpentier, he was the person to actually do it. So there is a face-off between MIT and University of Berkeley for the patent. And in fact, now the patent is held by MIT. But Doudna and Charpentier were awarded the Nobel Prize. So this is causing, this raises the question of whether every one of us will be, if we are also having such kind of cancers, will we be able to afford it? Will we be able to afford it for our children? And will it be accessible? So accessibility is another thing. So only the rich, if they tend to increase the money of such kind of applications because of patents, will the most general population be left out? So those are ethical questions which are have to be answered. And the future of genome editing is going to be, so everywhere you're going to hear it from now on. I am certain about that, because the way we perceive medicine, the way we do agriculture, the way we do many of our research in biology is being changed and revolutionized by CRISPR. And you can expect personalized therapies, that is customized for you, just one person, based on your genome. So those kind of medicine is going to come. It's already in the process. And the different kinds of advances on this technique is happening as I speak. From 2012, when I joined for PhD, by this time, almost 1,000+ papers have been published just on improving the efficacy of Cas9, right? So that is how it is. And there is another thing called so these people who are you see over here, only Charpentier and Doudna were the persons who got the Nobel Prize. But as I told you, Barangao and Howat, as well as this guy over here, they all published just 2 months back. I mean, after Doudna, and they could not receive the Nobel Prize. So what has happened was that they had initially sent an independent study they had done. They had sent it, but their review process took more time than Doudna's. So they were not able to publish that. So there is an issue of first reporting first. So that is something which as scientists who are over here, who are interested in science, we should all think of. But even though that is the case, we should all acknowledge the fact that this history of CRISPR, which I told you about right now, it shows that individually as scientists, as single people, we may not be able to do much things. But when you do them when you stand on the shoulders of giants, as Isaac Newton told, then we'll be able to accomplish more than what we can do individually. So with that, I'll thank KFRI for giving me an opportunity for this, and thank you, everyone.